I don't want to scare anybody, especially myself.
Okay, we're rolling. You go ahead and talk to them.
Okay. So can you tell us a little bit about what the guys are doing here and the project they're working on?
Yes, we have a U.S. Forest Service grant. We're working on improving the national biomass equations. There's 6 geographic regions in the U.S. Forest Service. We're the North Central region. And we have a U.S. Forest Service scientist and an MSU scientist paired together working on the biomass equations. They're getting a little old. The Forest Service has forest inventory plots all over the country. They follow the growth, the species of trees that grow, their health, their condition, and their biomass. And it's important to know biomass because then you can figure the carbon. And today everybody is interested in how much carbon we have sequestered in our forests. What they're doing is we're charged with, our crew is the biomass crew for the lower peninsula of Michigan, parts of Ohio and Indiana. We're collecting samples from about 7 or 8 different species of trees, 2-inch increments, trees from 2-inch all the way to 32 inches in diameter. What they do is they go in the field, they measure the trees on the ground, which would be the system you would use after we have all the equations improved. Then they cut the tree down, remeasure everything on the ground, cut the tree into sections, weigh each section of the stem. For instance, Mickey is working on a section here. This is 90, how many feet up on the tree?
Branch 2.
Oh, that's branch 2. That's amazing. The other one was 96 feet up on the tree. This section from a 27-inch tree is 12 foot up on the tree. We weigh everything green, including the stem, branches, and twigs with leaves. We then separate the bark from the wood, weigh the wood green, the bark green, the branches green, and the leaves green. Oven dry everything to get rid of the moisture. And then we weigh everything dry. And then Dr. McFarland takes all the data and he figures out the biomass equations that fit each species of tree and size class.
And what species are you looking at? You said 16.
Last year we started with red maple. We got our feet wet. We did it very intensively. Tried 3 different measurement types of instruments. We did every leaf on every tree. Now we just take subsamples of each branch, and we're now doing tulip poplar. We just finished. We're doing, these are white oak. Then we're going to do red oak and American basswood and American beech. We happen to have a harvest ongoing at Russ Forest, working with the Forestry 420 Special Studies class. During that harvest, some of the beech trees will not be cut, and we selected them for the biomass study. So we'll follow that harvest, cut those trees, and check the biomass of those.
And why is it important to someone living here in Michigan with this type of research? How might it impact them?
It would impact them because we know what is in our forests. Our forests are our health. And the forests, what they do is they sequester carbon, and we're putting more and more carbon in the atmosphere. The trees are sequestering that. How much they can sequester and how we can use that wood fiber for energy and heat and keep it in a closed system instead of adding new carbon to the atmosphere is important. This ties in with also with our energy plantations, our chip study that we saw earlier. And we'll see a harvest operation where green chips are actually going to the MSU power plant.
So you're studying species from all over the state then?
In the North Central region would be, think of Michigan, Wisconsin, Indiana, Ohio, probably Minnesota too.
Okay, great. Anything else you think would be important to touch on? I mean, we could talk about the importance of having a facility here that enables research to be done that might not be able to.
One thing is, where do we get all our samples? Or where do we find all our trees? Right now, we're stationed This is an Ag Bio Research Center, Kellogg Experimental Forest. We manage 7 different properties. Another one, Fred Russ Research Forest, is an Ag Bio Research Center. We work on all those properties doing research projects with all the researchers on campus. And then in this particular project, we search trees of these species. We get the American basswood at the MSU Tollgate Education Center. Friday, we're going to go to Tollgate, Trevor Nichols in Fenville and look for species there. We got the tulip poplar at Fred Russ Forest, the white oak and some of the red oak at McCready Reserve in Jackson. We found some white oak and red oak in Rogers Reserve, also MSU property. So we will also be going to Lake City to look for trees for specifically hemlock and white pine there. When we exhaust all the MSU properties, then we have permission to go into DNR property and get samples. So having all the MSU properties located throughout the state's ideal. And with a core crew of these 4 people, we can consolidate all the samples here, and then this is our main lab to process the samples.
When do you expect to have these equations? When will they?
That will be totally up to Dr. McFarland.
Right.
We're the field people. Just to collect the samples, we figure this is done from June to February. Last year we only did 1 species because we did it really intensively. This year, I think we're only going to get 3 species done. No, we may get 4. We have tulip poplar, red oak, white oak, and maybe American basswood. We'll have another year, maybe 2, just collecting samples.
Wow. Any idea what the weight of all the samples? I can't
Sam?
imagine.
For some of the red maple.
Well,
It's gotta be.
when they weigh individual samples, like
Yeah.
4-foot sections on the stem, what's the heaviest 4-foot section so far?
900.
900 pounds.
And that's
So-
4 feet
4
of
feet.
a 100-foot tree.
I do not know what the total weight of the tree is. We should know that because they've picked up the whole tree piece by piece.
Well, Neil said that 30-inch red maple is over 18,000 pounds.
Okay, our big 30-inch red maple, 18,000 pounds plus.
We had everything in here.
That's where we did every leaf, every twig. It's very intensive. But Dr. McFarland told us the first year would be really intensive, and then we'd back up and just take subsamples. So we know what's coming.